Assalamualaikum warahmatullahi wabarakatuh and a very good day. We are from Group 2 and we'll be presenting on the same topic, that is the African swine fever, but on the global context. So this is the outline of our presentation. We'll bring you through the introduction to ASF and then we'll talk on how the disease affects us and the world, the preventive measures and the mitigation strategies. Lastly, we'll cover a bit on grant funding and future approach. What is African swine fever? So basically, ESF is a contagious viral disease of domestic and wild pigs, and it is listed as notifiable by the World Organization for Animal Health. ESF is a deadly virus disease because death can occur within 7 to 10 days after infection, and it has been reported that sudden death may also occur. The causative agent of ESF is, of course, the virus, ESFV, and pig species are the only susceptible animal population. ESFV is well known as a virus with a significantly high environmental resistance, and it is large, enveloped with double-stranded DNA that can replicate in the cytoplasm. So, African swine fever was first identified in East Africa in the early 1900s as a disease causing high mortality in domestic pigs. But then research found out that warthogs and bush pigs, as you can see photos on the right is warthog and left is bush pig, could be the source of infection. And these hosts, along with a species of soft ticks which live in warthog burrows or pigs housing, could be persistently infected with ESFV without showing signs of disease. Like I mentioned before, ESFV is highly environmental resistant. Research has shown that the virus can shed in large quantities in the blood, and it can survive for 15 weeks at room temperature, months at 4 degrees Celsius, and for a very long time when frozen. In raw meat, it can survive for more than 3 months, and even the infected pig's feces and urine are also infectious. Meanwhile, the carcasses of infected wild boars could also play a major role as ESFV reservoir, and it can help to locally maintain and spread the virus to the other wild boar populations. Simply to put that any contaminated food waste, material, vehicles or equipment, and also the person who involved with the equipment could represent a source of infection to ESF-free countries. Even in countries where outbreaks have already been reported, a higher level of environmental contamination can pose a higher risk of further outbreaks. Actually, ESF is not considered a new disease. According to research, ESF was first diagnosed in Kenya in the early 1900s, and the further spread was limited to Central and Western Africa until it was discovered in Portugal in 1957. From a period of 1960 to 1986, ESF spread was detected in Spain, France, Italy, Sardinia, Belgium, the Netherlands, as well as in Caribbean and also South American countries. But then in 1990, all of those countries except for the island of Sardinia were successful in eradicating the disease. However, ESF made its comeback to Africa in 2005. In 2007, the disease was confirmed in the Republic of Georgia, and from there, the virus gradually spread to the neighboring countries. And then the first occurrence of ASF was reported in European Union in 2014. In August 2018, the virus leapt to China. This represents the first occurrence of ASF in Asia. In September 2019, the first occurrence in Oceania. And in July 2021, the disease reappeared in the Americas after an absence of almost 40 years. And lastly, since January 2022 until recently, ESF is present in the African, European, and Asian region.
Now let's look into how ASFV can be transmitted from wild to domestic pig. Transmission of ASFV can be categorized into three modes, which are direct, indirect, and through tick vector. Direct transmission has been mainly demonstrated by the contact of infected pigs via infectious body fluids, nose-to-nose and bodily secretion, as well as aerosols of a short distance in between pens. In indirect transmission, it involves ingestion of contaminated feeding, such as feeding on garbage containing infected meat. ASFV is known to be highly resistant as it can remain infectious up to 36 months in uncooked pork products. Next, we have biological factors such as tick and warthogs as the agent of indirect transmission. For mites, object or material, such as premises, vehicles, implements, and clothes, also contributed to the indirect transmission. The third category, which are also one of the indirect transmission, is a tick vector. ASFV can replicate in ornithodorus, and they are the most common vector of the virus. It can be transported mechanically to susceptible pigs by biting or through biological process, which are transtadial and transovarial. Now let's look into the diagram. Through this diagram, we can see on the left side how tick carrying virus from sylvatic cycle entering domestic cycle to infect domestic pig. And on the right side, we can see tick-to-tick transmission, which are transovarial and transtadial. Transovarial, ovary, contain ovary in the name, means that the virus is being passed down from parent to offspring via ovary. While transtadial, star means stage, meaning that the virus is being passed down during life cycle or life stage. In transtadial, the virus will infect the tick vector as a larvae, and the infection maintained when it morphed to nymph and later developed as an adult. African swine fever is a highly contagious viral infection of pigs with a mortality close to 100%. It has been a growing concern as we speak at the moment to those affected countries, including Malaysia, and we must each do our part to address the threat before it's too late. And this is why understanding the mode of transmission will help to break the chain of spreading the ASFV.
Clinical signs of ASF. Firstly, the virus takes about 5 to 15 days or maybe less than that as an incubation period before the clinical signs start to appear. This period actually varies depending on the virus mode of transmission. It could be 5 days or less when the virus enters the body through tick bite, while it takes 5 to 15 days after direct contact with other infected pigs. After the incubation period, the clinical signs of the disease start to appear on the infected pig with different forms of severity. Uh, it could be either peracute signs, which is the most severe form of the disease, acute, subacute, or chronic, which is the least severe form of the disease. Actually, this staging of the clinical signs depend on the strain of the virus because there is different strains of the virus. It could be highly pathogenic strain or less virulent strain. The peracute signs mostly characterized by sudden death, while the acute signs could include high fever, which is usually 45, 41 degrees or more than that, lethargy, weakness, anorexia. Hemorrhagic signs include erythema, red skin, particularly in the pinna of the ears. Respiratory signs include dyspnea, nasal and conjunctival discharge, abortion in pregnant pigs. The subacute signs is similar to the acute signs but less severe, and pigs that or those develop this kind of signs usually die or recover within 3 to 4 weeks. The clinical signs usually they are not specific signs. They could include intermittent fever, appetite loss, depression, joints swelling, coughing, diarrhea, and occasional vomiting.
What are the effects of African swine fever globally, and how does it influence Malaysia? According to Food and Agricultural Organization (FAO), in 2013, as much as 113 million tons of pork carcasses was produced globally. The region of Asia dominates the market with a whopping 57% of global production, followed by the Europe and Americas with 24% and 17% respectively. The global pig supply chain is also the largest meat contributor in the global meat production, with 37% in 2010. This amount is projected to be increased 32% by 2030 alongside its demand. Economically, we can see that China is the world's largest pig meat market, contributing 45% of global production and consumption in 2018. However, during the outbreak of ASF in China in 2018, we can see that the output fell by 20% in 2019. The decrease of production with a steady and high demand creates a situation where the government are forced to import more from other countries. This will in turn increase the global market price of import, and it is threatening the global market value of food price. Other than that, we can see that the decrease of pig population in China creates a surplus of feed and decreased feed demand, which usually comprise of soybean and cereals. Since China does not need to import the feed, this will further contribute to the volatility of grain price. In a more individual scale, ASF impact the hunters by encouraging them to hunt more wild boar but with precautions and proper guidelines in order to prevent the spread of ASF from wild boar to domesticated pig population. Clear communications must be established with the authority body such as World Organization for Animal Health with the hunters to prevent the outbreak and to encourage the hunters to report any incidents without facing the stigma of carrying the outbreak. However, the effects on farmers are more devastating as they involve losses of capital. It was reported by Babalobi that the farmers of Ibadan in Nigeria suffer losses a total of 941,491.67 US dollar due to the death and culling of the pigs. It is reported that the World Bank compensated the farmers with the National Livestock Service Project Fund, but only 300,000 US dollars were allocated. ASF did not affect much of the environment, but the aftermath of it does predict a significant impact towards the environment. OECD reported that the pig meat production are expected to recover, and from Woon Wong et al. Suggested to do so, the pig industry must be transformed and increase its production in large scale. As we know that the livestock sector is already giving a major impact to the environment, using 35% of total arable cropland and 20% of water. Other than that, it is known to be one of the procedure to dispose the infected carcasses with the burning of flammable toxic material, but it is done under the supervision of competent authorities. It is known that the ASF virus is a different disease from the swine flu, and it is belong to the different family. ASF does not infect human. However, there are research by Loh et al. Found that ASF virus-like sequence were found in human serum, but it is unlikely that there would be a major implication towards human health. Let us look at the impact of the ASF on Malaysia in global scale. According to OED, Malaysia exports 7.39 million US dollar, making it the 43rd largest pig meat exporter, and 43.2 million US dollar, making it the 51st largest importer of pig meat in 2020. ASF were detected in Sabah in February 2021, and all 14 samples from 3 different locations were found to have ASF virus. Sawah Hanif and Nazir from Ministry of Agriculture and Food Industry reported that Malaysian pig industry, which are valued at 5 billion ringgit, will face losses comparable to previous outbreak of Nipah virus. Other than that, in order to curb the outbreak, import bans and restrictions might be needed to take place. However, this will lead to dissatisfaction in exporting countries, and they may take a further legal action against Malaysia under the World Trade Organization.
How do we prevent ASF? Culling is still the gold standard as for now. And what is our understanding about culling? So, culling is actually reducing the population of the animal by killing them. But there are a lot of things to be considered, to be pondered about when we plan to do this. For instance, timely compensation, environmental damage. Where are we going to dispose the carcass in large amount? Social acceptance. Can people accept the fact that we are planning to do culling? And cost-effective disposal procedure. As for now, Vietnam actually came up with a study. They combined with US, so they combined with the United States Department of Agriculture, USDA's Animal Research Services, and they came up with a vaccine that had an immunity lasting for about 6 months. It was actually done on partially killed infected pigs. The study was done on partially killed infected pigs. However, has been temporarily suspended after dozens of pigs were that was inoculated with the shot died in August 2022. There are several things that we can actually do to prevent ASF. For instance, provide rapid detection tests in local farms. For example, make it available for farmers to have PCRs so that there can be early detection. It can be achieved and the farm's biosecurity is increased. Besides that, we should implement appropriate import policies and biosecurity measures for ASF-free countries. By this, I actually mean that implementation of appropriate import policies and biosecurity measures for ASF-free countries, ensuring that neither infected live pigs nor pork products are introduced into ASF-free areas. Then we should also impose strict regulation at immigration regarding pork products. Awareness is one of the main preventive measures. Farmers should actually have the awareness on how serious this issue is, and they should be able to declare any suspicious cases. But to do that, there should at least be a certain form of compensation towards the farmers, because they are going to be at a loss. So we should be able to come up with a budget whereby we can give them the compensation, so that they would have the awareness. Okay, we should report the problem. We should be able to come up with training whereby we train the farmers in the sanitation of the farm, the importance of sanitation of the farm. As for Malaysia, we actually have increased the precautions to avoid the entry of the virus to the country, and as published by the Ministry of Agriculture and Agro-Based Industry of Malaysia, they have not only forbidden the import of pigs and pork products from affected countries such as China, Vietnam, Poland, Belgium, and Cambodia, but they have also expanded the prohibition to Thailand, as Malaysia's border Thailand to the north, and therefore Thailand was also a contaminated area. And also, the Malaysian Inspection and Quarantine Services, MAKIS, are strengthening the controls of the international airport to avoid the entry of products from countries affected by ASF in travelers' luggage, as well as through the land border with Thailand. Likewise, the control to avoid the illegal imports of products from unauthorized sources are being increased.
Okay, now let's move on to our mitigation strategies to Malaysia. According to the latest update of African swine fever (FAO), Malaysia also had been affected by the ASF virus, with the latest case is in Perak. So, what are our strategies to mitigate the spread of the virus in Malaysia? The first strategy is to increase strict enforcement of biosecurity at the country level. This means that Malaysia should temporarily ban import of pig livestock or pork products from affected country, and maybe perform risk analysis before approval of import permit. Malaysia also could maintain strict import policy controls. This can be done by establishing cooperation with agencies such as Custom and Marquis to inspect and confiscate of pigs and pork-based products that brought into the country without valid permit. Imported live pigs also need to be quarantined and monitored for 14 days by Marquis at the animal quarantine station before the permit is issued and the livestock is allowed to be transported to the destination state. The second strategy is to improve information transfers with international forum on risk and mitigation of ASF, as well as having international engagement and collaboration with affected Asian countries. Therefore, Malaysia will have the update on the latest ways to mitigation the virus and avoiding further loss in the industry. The third strategy is to take immediate action to the infected farms of pigs. This can be performed with cooperation with Department of Veterinary Service to stamping out the infected or suspected pig in the farm or premises. The stamping out should be done preferably without involving slaughter or bleeding and considering the animal's welfare without the risk of further spreading and polluting the environment or premises during the process. Carcasses must be properly disposed to avoid becoming source of infection through scavenging animals. They could either be buried in deep hole or burned. A proper compensation to the pig owners or farmers should also be made to encourage them to report any further suspected positive case and avoid illegal dumping of the carcasses and then lead to further spread of the virus. Strict enforcement of biosecurity on affected farm also should be done. This action is implemented by cleaning and disinfect the whole farm, including equipment, clothes, and transportation used. After the process had been done, whole premises, including equipment, must be sprayed with acaricide to kill pig ticks. The premise also must not be in use, and new animal only can be transported in after 40 days. Surveillance should also be done and continued for 14 to 48 days after the disinfection process of the premise. Proper surveillance also must be done to the nearby premises in the area to identify any other infection. The fourth strategy to mitigate ASF is to increase awareness to the farmers and other stakeholders. As for the farmers, they should be aware of the clinical symptom of the ASF infection for them to make early report to the DVS. They also should know the ways of transmission and the needed biosecurity action so they would not infect the other farm or premises as the virus is very resilient. The other stakeholders that might be involved in these strategies is custom and markets in the import of pig, livestock or pork-based product, airline services for the in-flight announcement of prohibiting carrying pork-based product in the flight, airline services and port authorities for waste management and disposal food and kitchen waste from airport and ports, and perhelilitan for wild boars population and control activities.
Okay, let's proceed with grant and funding in the research. So far, there are many grant and funding had been given since the African swine fever is identified. As you can see here is the timeline for the grant and funding in these recent years. In 2018, European Union Horizon 2020 Research and Innovation Programme has funded an international partnership called DEFEN, where academic, industrial, and governmental organizations working together. There are three aims that DEFEN want to achieve. The first one is to develop practical tools to prevent further ASF spread. Then to compare the effectiveness of the current control measure. And the special one is to develop a peptide-based ASF vaccine against the genotype 2 ASF virus strain that currently circulating in Europe. There are three grant and funding given during 2019. The first one is from United States Department of Agriculture and National Institute of Food and Agriculture. They form a team called ASF, NIF, and NF and have three main objectives in their research. The first one is to assess the pig contact network within the region.
to explore tick involvement in ASF virus transmission and also to assess ASF seroprevalence and viral diversity in the sylvatic and domestic cycle. The second fund given in 2019 is by Foundation for Food and Agriculture Research to Pipestone Applied Research Team. Their main aim is to develop a mitigant, which is an additive that deactivate ASF virus and other viruses. Basically, this mitigant is added directly to animal feed. And then Foundation for Food and Agriculture Research, together with National Pork Board, awarded a fund to Kansas and Iowa State University in 2019. These two universities have two different aims in their research. For Kansas State University, they want to understand how ASF virus survive and continue to infect other animals in various environments. While for Iowa State University are focusing on how to identify foreign animal diseases at low prevalence using oral fluid sample. In 2021, United States Department of Agriculture had given a grant to Swine Health Information Center, but the research is being done by Biostone Animal Health together with Canadian Food Inspection Agency and National Centre for Foreign Animal Diseases. Their research is on ELISA for ASF and to determine the diagnostic sensitivity and specificity of the ELISA using a pig serum sample with known ASF infection status. In March 2022, United States Department of Agriculture and National Institute of Food and Agriculture has given a fund to Kansas State University. Their research will be focused on rapid and early detection measures for ASF through antibody testing and on how to reduce the risk of imported feed ingredients serving as a route for introduction of foreign animal disease, including ASF. The latest one is a fund given by 4 different organizations, which are Foundation for Food and Agriculture Research, United States Department of Agriculture, Agriculture Research Service, and Plum Island Animal Disease Center to Genvax Technology Corporation. So, Genvax Technology Corporation will develop a self-amplifying messenger RNA vaccine for ASF. Lastly, in the future, there will be more and more grant and fund given to anyone or any team that doing a further research related to ASF. The results from this research will become the future approach to prevent and further spreading of the ASF and to stop this ASF epidemic globally.